Okay, 3.3, 3.4. I'm assuming you have read it. Always seems kind of an obvious thing, like duh. You're doing a section in the textbook, you probably should read that section, right? So, yeah, reading is a little bit of a lost art. If you find yourself that you're not all that excited about reading, I need to encourage you that that's a skill you really want to learn. It's an academic art that really will take you far, right? So reading is wonderful. This particular package is more of a lab package, and the only reason I'm actually pointing it out to you is that, yeah, you need to dissect a flower or two, and you need to dissect a seed or two. The actual lab procedures are on page 98, 99, 100, and 101. Your package contains the lab write-up material, but make sure you read this page, because on this page is a lot of important information about what you might need to attach to that package, because not everything in that lab is included in the questions, right? Okay, before getting on to the flower structures, their functions and whatnot, just a quick reminder that you have a website that goes with your textbook with a whole lot of web links, multimedia. These are the multimedia links for Chapter 3. 3.1 includes a whole bunch that really are important for you to see. Don't forget the password for it is on your course outline, or you can talk to me. We're, of course, in 3.3 and 3.4, which means you're looking at flower parts. When you get to seeds, you're looking at the monocot life cycle, comparing it with, say, a dicot. I'll show you the flower part one here and the monocot life cycle, but I'd encourage you to check out some of the rest. As your textbook, of course, gets older, some of those links might be dead links by now, but, the ones that work are probably not too bad. So this is a flower.
The female reproductive parts are at the center of the flower. A cherry blossom has a single carpel. The upper part of the carpel is a sticky stigma where pollen lands. It is elevated on an extension of the ovary wall called the style. The lower part of the carpel is the ovary. Here, eggs develop from megaspores, fertilization takes place, and seeds mature. Stamens are the plant's male reproductive parts. The stamen consists of an anther and a filament. An anther is divided internally into pollen sacs. Within these chambers, haploid microspores form and give rise to the male gametophytes called pollen grains. The sepals and petals are the non-fertile parts of the flower. The sepals are the outermost leaf-like parts. All the sepals combined constitute the calyx. The calyx surrounds the petals. All the petals combined are called the corolla.
Okay, the flower, as I mentioned, is in fact a sex organ, and the sex organ contains both male and female sexual structures. I think it's only fitting that we go with blue for the male parts, and collectively the male parts are called the stamen. Stamen. And the stamen is made up of the anther here, anther, and also the thing that holds the anther up, which is the filament. So stamen is made up of the anther and the filament. You're going to notice on the anther, if you have a real flower in your possession or see one in the near future, these little dots that usually fall off on your mother's tablecloth and stain them, or if you stick your nose in a flower, you wonder, okay, what in the world just got on my nose because it stains. That is called pollen, and pollen is the male structure that contains the sperm. During fertilization, the sperm actually ends up on this structure here and travels down this long tube all the way down to the female structures, the ovary, where fertilization takes place. I suppose if we're going to go with sexist colors here, we better switch to like pink for, no, pink for the girl parts, right? So that looks like pink to me. Sure, let's try that. Collectively, the female parts of the flower are called the pistil. What just happened? What the heck is that? I don't like the look of that. Let's just experiment. let's see now. I think I did something wrong. So we have to go for like pink, but not nearly as thick as I had last time. So let's go for like that. Okay, try it one more time. Female structures collectively are the pistils. And the pistil is made up of this tall structure that I just mentioned where the sperm will actually travel down, called the style. And it has a sticky stigma at the top. We'll just call it stigma. So the stigma, it's kind of sticky. You want pollen to land there, so you'll often see insects, their little feet all covered in pollen from another flower, landing on another flower, and that pollen is left behind, and then, as I said, moves down, 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 down into the ovary. When it's in the ovary, it actually meets these little eggs called ovules and fertilizes them. So that's where fertilization takes place, in the ova. So then there are parts of a flower which are neither boy parts or girl parts. So I'm thinking we should just go for green for those. What do you think? They would be the petals. Not that they're not important. They attract insects. They also, look good. And then the sepals, these little structures. Not all flowers will have them. Depending on which flowers you're dissecting, yours may or may not have them. Collectively, we call the corolla the collection of all of the sepals. Okay, it is one thing to be able to label a diagram of a flower, but it's another one to be able to give the functions, right? So make sure in your particular package that you have identified these functions. I believe in your package we give you the functions, but we didn't name the structures. So for instance, the petal is the colored leaf-like structure. Stigma is the top of the tube-like pistil, receives a pollen grain, which will then travel down the style to the ovary where the eggs are contained. I think you can read, right? So I'll leave that on the screen here for a second, and you can, just make sure you know both structure and function.
A lily flower is a specialized reproductive structure. Female gametophytes form in ovules, and male gametophytes form in pollen sacs. Inside the ovule, meiosis is followed by 2 rounds of mitosis without cytoplasmic division. This produces a single cell with multiple nuclei. Cytoplasmic divisions result in a 7-cell female gametophyte. Inside each pollen sac, meiosis and cytoplasmic divisions produce haploid microspores, which develop into pollen grains. After pollination occurs, the pollen grain germinates and a pollen tube grows toward the ovary. The tube contains 2 sperm. It is the mature male gametophyte. When the pollen tube reaches the ovule, 1 sperm fertilizes the egg. The other fertilizes the endosperm, producing cell. Double fertilization produces a seed in which the embryo sporophyte is surrounded by a nutritious endosperm. The seed germinates and grows and develops to produce a mature sporophyte.
Yes, the seeds. Seeds are miraculous little beasts. If you think of taking a tiny little seed, sticking it in the ground, and potentially coming back hundreds of years later and seeing a gigantic redwood. Seeds, as I've said, feed the earth. They're important, and they show amazing design and function. Suggests to me that there was a designer that probably designed them. Anyhow, we're looking at 2 types of seeds. Dicots stands for dicotyledons. Cotyledons are just seed leaves, those leaves that pop out of the ground when your bean plant begins growing, or they're monocots, which stands for monocotyledons. The difference really is in those seed leaves, when you have a bean and you plant it, you get 2 seed leaves that pop out of the ground after germination. When you have a seed like corn, which is a monocot, you're going to get 1 seed leaf that pops out of the ground. So you need to know the structure and the function. Structure A here is what we will call the epicotyle. Epicotyle. And those will develop into the plant's stems and leaves. B is the seed coat. Seeds deal with amazingly harsh environments. When you think of the soil, you have very rough gravel, sand, insects, critters like worms that are trying to eat you. So seeds have a protective coat called a Tessa. Sounds like the electric car. That is really their function is to protect the seed. C would be the seed leaves themselves. And I don't like that popping up. Go away. Cotyledon. For some reason or other, still learning how to make this puppy work. I should send you to school to work on a tablet. Cotyledon. These are the seed leaves. And then we have something called the radicle, D, which grows into the root. Not radish, radical, that's a C. Radical, those will become the roots. So they have an interesting tropism or a behavior. They will always grow towards the center of the earth, whereas the cotyledons will always grow towards the sun. This is a positive phototropism, they say. This is a positive geotropism. Then we have structure E, which is the helium. And the helium, you don't really need to worry too much about, other than the fact that it was the point at which that beam was attached to the stem originally. Makes sense? Makes sense. By the way, that animation you just watched, again, there's more information in there than you really need, but again, it doesn't hurt to watch it. So let's compare the bean, the dicot, with the monocot, the corn. Okay, A is now the seed coat in the monocot. And again, somehow my poor little palm is resting on something that it shouldn't, because it causes bad things to happen. And again, the seed coat is there for protection. B is what's called the endosperm. And the endosperm has an important function where it's stored food. So until a plant can start photosynthesizing through its leaves, the leaves haven't developed yet, it's important that there's enough energy potentially in there in the stored food. So that's what B is doing, all of this here. By the way, when you do the seed lab, that stored food is going to turn up as starch. Seeds contain nutrients, that's why we eat them, right? So you eat seeds, that starch gets broken down into glucose. So I'm going to write that down here. Stored food, that stored food is starch. And there's a test for starch, which is iodine. If you put a drop of iodine on an open seed, if there is starch present, you are going to get little black dots. So I'm going to show you where you get little black dots. Boop, 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 boop. Wherever the stored food is, you will begin to see these blue-black changes. Blue, black. So if you notice when you stick iodine on a seed, the change of color into blue-black, you'll notice there's plenty of stored food there. So C happens to be, what do you think? The seed leaves again, cotyledon. And D itself would be potentially the leaves. And that's of course important for photosynthesis when the plant begins to make its own food. And then E is the young plant itself, the embryo. Make sure you read this. Nice summary of the differences between the monocots and the dicots. And yeah, I think the sooner you can get at that lab, the better.